Hi everyone, and welcome to our first Animal Behavior Live seminar. So today we are welcoming Dr. Yusuf Abdullahi and Dr. Fiona Mumoki from the University of Pretoria in South Africa. So they are going to present the social parasitism by an African honeybee clone. And just to make sure of the format, so the talk will last 45 minutes and will be followed by 15 minutes of questions on the YouTube chat. For those that won't be able to attend the live streaming, the video is going to stay on YouTube forever, so you can watch it in replay. And you can also ask your questions on Discord directly to the two researchers. So they will answer your question whenever possible on this platform. So now a quick presentation. So Dr. Abdullahi main research interests are insect behavior and chemical communication, pest and vector management, the evolution of asociality, cultural entomology, and analytical method development. Dr. Abdullahi obtained his PhD in 2010 in entomology at the University of Pretoria in South Africa. He then moved to Nigeria as an assistant lecturer, and he also pursued with a postdoctoral fellowship and several research fellowships in South Africa and Germany. And since 2018, he is now a senior lecturer in the Department of Zoology and Entomology at the University of Pretoria, back in South Africa. Now, Dr. Fiona Mumoki is a postdoctoral fellow. She works in the same laboratory, so the Social Insects Research Group at the University of Pretoria. Fiona was born in Nairobi, Kenya, where she also undertook her basic education. She received her bachelor's degree from the University of Nairobi in Kenya, and afterwards spent time carrying out a research internship at the International Center of Insect Physiology and Ecology, still in Kenya, focusing on developing molecular tools for diagnosis of honeybee diseases, earning a master's degree in genetics from Jomo Kenyatta University of Agriculture and Technology in 2014. In 2015, Fiona traveled to the University of Pretoria in South Africa to join the Social Insects Research Group, working with Professor Robin Crew, Professor Christian Pierk, and Dr. Yusuf Abdullahi in exploring the molecular basis of reproductive dominance in the Cape honeybee, earning her PhD in 2020. And she is now a postdoctoral fellowship in the same laboratory. So back to you, Yusuf.
Thank you very much, Natasha, for the nice introduction, and thanks to everyone for arranging this seminar. It is a pleasure to use this platform to tell you about the work that we do at the Social Insect Research Group at the University of Pretoria. Before I delve into the talk, I want to say a little bit about our university. So the University of Pretoria, it's one of the largest universities on the African continent, and it's actually the largest research-intensive university in South Africa. We offer around 1,400 study programs, and we have quite a high number of percentage of undergraduate pass rate and also employment rate. We also have a large alumni network, which is about 300,000. Back to our primary host, which is the Department of Zoology and Entomology, we were ranked the 50th in terms globally in the field of zoology and entomology, and then so we have multiple teams working in different interdisciplinary research at the department. So we have colleagues working on mammals, from the largest mammal you can think of up until the smaller one, and also those of us working on insect, looking at different aspects. So if you want to know more about the work that we do and that of our colleagues, you are free to visit the website. Now to our primary group, which is the Social Insect Research Group. As the name implies, we work mainly on social insects, so bees, ants, and termites. And for today's talk, we're going to focus more on one of our key models, which is the honeybee. So now our interests in honeybees are multifaceted. So one of the interests is looking at how eusociality is in honeybees in terms of how worker reproduction is controlled and how the queen controls the worker. So one of the key aspects that we work on, especially my interest, it's looking at chemical communication, where we use analytical chemistry to encode or decode chemical communication in honeybees. We also work on other pollinators and especially looking at how monocultures affect pollinators, both wild and honeybee pollinators, and also looking at nutritional aspect of pollination as well. In other words, what are the resources that are good for pollinators and what are not. Also, we also study other arthropods within the honeybee hive. Some of them include the pests like the small hive beetle or varroa or brawler, just to look also at communication in them, because some of them have evolved ways to eavesdrop into honeybee communication system, and that is why they coexist within the hive. We also work with colleagues in genetics and also in mathematics, developing mathematical models that will help us understand or entangle e-sociality in honeybees. So for today, myself and Fiona are going to take you through a journey, what I call a journey that is from our studies on the pheromones, that is from the chemical perspective, that is from the molecule level down to the gene level, and also the synthesis. And then later on, I will round up the talk looking with this question about what of other pheromone signals from other glands. The first part, which is what I will talk about, I will briefly usher you in. It's just a basic introduction into African honeybees and the mandibular gland pheromones, and then introducing our main model, which is the South African honeybees, especially the capensis clone, which you see now how unique it is, and then also show you how we were able to make false queens from workers of honeybees. Chemical communication in honeybees, or specifically in African honeybees, is very important. So if you take the case of honeybees in the colony, which is dark, the main means of communication is using olfactory cues. And these olfactory cues are from different exocrine glands. So I have highlighted here the main exocrine glands that have been studied and which will be form the basis of this talk. So one of such is the mandibular glands. The other one is the hypopharyngeal glands, which is usually only developed in nurse bees, and it disappears in foragers. And then we also have the Koshkonikov glands, and then the Nazanov glands, which is mainly secreting chemicals that or pheromones that are known to use in coordination. Here, it's very important and interesting, and it was noting that the Targal glands are only fully developed in workers of African honeybees. In other honeybee subspecies, the Targal glands are only present in the queen, so the workers don't have developed Targal glands. Now, so having said something about the exocrine glands and the different secretions there, now you wonder how communication is, because now you have these multifaceted glands that are continuously secreting chemical signals. So what are they used for, and what are the audience? So let me take you through a very simple representation of how chemical communication is in the hive. So at first we look at the queen here. One of the main pheromones that she uses, the queen mandibular gland pheromones, and one of its function is in the attraction of drones for mating when she goes out for mating flight. The queen also uses the queen mandibular gland pheromones to coordinate the workers. So one of these coordination is the bringing together of a retinue of workers around her, and also using the target gland also, which we found on to be complementary or work in synergy with the mandibular gland, because it has both primer and releaser effect. I will mention this at the end, and then also the Dufo glands. In terms of communication between the workers and the workers themselves, so here they use mainly cuticle hydrocarbons for nest mate recognition, and also use mandibular glands pheromones and also use secretions from the nasal glands for communication. On the other hand, the workers also communicate with the brood. So this is using brood pheromones that are released by the brood. This is now for the brood requesting for food and also regulation of foraging and others. There is also another channel of communication which has been often neglected. This is a communication now between the drones and drones themselves. So what we know more is about the communication of between the drone and the queen during mating. But of recent, there are evidence that show that the drone has mandibular glands that also secrete pheromones, and these pheromones are being used for drone-to-drone communication. So the main endocrine gland that we work on is the mandibular glands, and it is found here inside the mandibles. This is on the heads of the bees, and it is found in both mandibles, both the left and the right hand side. So this is how an exercised mandibular gland look like, and this gland has a very interesting chemistry. Even though there are more than 60 compounds, only 6 have been shown to actually serve and actively as pheromones. So one of the interesting chemistry of this gland is that these pheromones that have been shown to be active in honeybees are caste specific. So for the queens, it's usually the 9 fatty acids, and then including this ethanol and this hydroxybenzoate, while for the workers is the 10 hydroxydecyanoic acid. So how do bees get these pheromones, or how are they synthesized? So I will just go into this briefly because Fiona is going to give a detail. So the starting molecule is stearic acid, which undergoes esterification, and then it has these two bifurcated pathways. One is the queen pathway and one is the worker pathway, and they're different chemical processes from functionalization, beta oxidation, and the hydroxy oxidation group. So, but for the purpose of this talk, let's just mind the letters, not the name. So wherever we see 9 and it's in purple, it's a queen substance. Wherever you see 10 and it's green, it's a worker substance. So we're not going to mind these 2 queen substance for now. So what are the functions of this mandibular gland pheromone? So mandibular gland pheromone is one of the pheromones that has been shown to have both releaser and primary effect. Releaser effect are those behavioral responses or behavioral effect that pheromones exert on the receiver. So if we take the queen signal, for example, in terms of releaser effect, you have the retinue formation of retinue, clustering during swarm, and also drone attraction during mating. The primary effects are those physiological effects that pheromones exert on the receiver. So one of them is the stopping of queen rearing in the presence of the queen, and then stopping of worker reproduction. It also has a releaser effect in eliciting worker activities such as comb building, brood feeding, guarding, and foraging. So it's a total control pheromone, if you put it like that. So having introduced the mandibular glands, now I have to take you through the honeybees of Africa. So in Africa, our honeybee subspecies or species is the Apis mellifera, which has more than 20 subspecies known, and half of which are present on the continent endemic. So depending on which literature you look at, it's said that we have between 10 to 12 subspecies of African honeybees. And one interesting thing about our bees is that our bees are diverse. You find them in different biomes on the continent. So let me take you through this, walk you through this presence. So down here in South Africa at the Cape, we have the capensis bees, the Cape honeybees, which are only endemic here in the Cape region. And when you come up to our region here in Pretoria, north, you find the scutellata. The scutellata is widely distributed because its ranges expand up until East Africa. And at the subspecies that is very common on the continent is Apis mellifera adansonii, which is known as the West African bee, but it's actually present from some northern part of Southern Africa up until the coast of West Africa. When you go up in the desert, you have the Saharensis, and then you have the Emenetica. On African mountains, you have a localized or endemic subspecies known as the monticola. So in terms of diversity, our islands also have endemic honeybees like unicolor, it is found in Madagascar. So I've told you about the mandibular gland pheromones of our bees or bees in general. So the impression would be that we know a lot about this, but this was not actually the case. So some years ago, whilst doing a literature search, I discovered that we only know from about 2 or 3 subspecies. And then from those subspecies, most of what we know are actually from workers that are queenless. So that ignited another research question, and then made me to, together with colleagues, to look at this and see what we know or what are the differences, because we know about mainly about 2 subspecies, the Cape honeybee and the Scutellata. And then we embarked on this ambitious project where we wanted to sample all honeybee workers on the continent under queen-right condition, and then look at the mandibular gland pheromones to see what we will find in there. But with such a project comes a challenge, because what the method being used mainly is that you have to collect bees freshly and then exercise the gland, and then you analyze the pheromones. But this would not be feasible when you have to go around the continent. So we had at that time already a method which we have optimized, where it's very simple. What you need is you just need to collect your bees, sample the heads, put in a solvent. As soon as they are in an appropriate solvent, you can then analyze it in the lab. And then this method, we have a method of sample preparation, which is target specific. Since we know the pheromones, we actually go for them and then we analyze them. And then this was what we found. So remember the 9 and the 10, and the purple and the green. So this is now capensis from here, the red. So what we found here is that the capensis workers actually have pheromones that are more queen-like. If you take Intermissa, Saharensis, and Scutellata, they are mainly more worker-like, with some traces. And then looking at Adansoni, you have someone which is in the middle here, which has the precursor of queen, that is 9HDA, and also 9ODA, even under queen-like conditions. And then for unicolor, it's also something similar, but it's more worker-like. So this is quite intriguing, and it made us to think further in terms of conserving and preserving our honeybee subspecies, and also try to make sure that if we want to preserve this diversity, beekeeping should be mainly using localized subspecies. But then this is quite intriguing, because later on, if Fiona discussed the pathway, and if you recall that these are 2 different synthetic pathways. So what is happening, that you find a bee is sitting here while it's supposed to be a worker. There must be something that is happening. So we tried to look at this further. So now back to our models, which is the Cape honeybee. So the situation in South Africa is that if you remember here, we have the Cape honeybee, which is endemic here, and then you have the Scutellata in the northern region of South Africa. So this was the situation before 1990. And then here we have a region where you don't have honeybees, or mostly you find the hybrids, but this would say the capensis cannot move into this region like of Scutellata. But one interesting biology of the Cape honeybee is that it has some unique characteristics. One of the unique characteristics, as you've seen earlier on, is the presence of a queen pheromone. It can also lay diploid eggs, and these diploid eggs can give rise to female workers. This is not the norm in honeybees or in most haplodiploid insect, where you find that diploid eggs are only produced by one specific caste, which is the queen. But in the Cape honeybee, workers can also lay eggs which lead to diploid. And then they do this in the absence without fertilization. So they have these developed ovaries, and they also have always have a presence of spermatica. But since there is migratory beekeeping in South Africa, so there have been movement of bees from the Cape up in the here. One movement in 1990 resulted in something that we call the capensis calamity, which we're sitting with now. So situation after 1990. So as a result of this movement, we have now one clonal lineage of this wasting honeybee, which now Cape honeybee, which becomes parasites in the hives of Scutellata honeybees. So if you recall that these are bees that can develop diploid females and also have pheromone, then which means that it has an undue advantage on other honeybees, and it can actually use of it. But the situation after 1990 was not the first time that there were outbreaks of the capensis. So there were reported outbreaks earlier in the early 1900s and also in the early 1980s, but these were not as famous or did not persist as that of the 1990s. So some estimators at that time was that there was losses of more than 100,000 colonies annually since then. And then surveys most recently showed that colony losses were between 30 and 46%. And these losses were mainly in the Scutellata region. And these losses were from migratory beekeepers, which they lost more than 35% in comparison to stationary beekeepers. So you wonder, how can another bee become a parasite to the other? So this is what happened. So when you find a capensis clone getting into a hive of Scutellata, what it does is it masks itself, and remember it has pheromones that look like a queen, so it gets the workers of the resident hive to tender to it, and then it lays egg, and before you know it, it is replicating itself as clones. Then there will be a time where the resident's queen will die. We suspect that it's because she's not receiving the care that she ought to receive, because now there are many other queen-like things, if you like, queen-like bees to be tended for. And as soon as she is lost now, the resident workers dwindle, because these capensis clones do not forage, so they don't contribute to any hive activity. When you have here an intercept here, and then this dwindle, and then they get to the peak, and then they also dwindle. So at this time, the parasites now leave the hive and then seek other hosts. So if you taking back to this map of this distribution, so if you look here, this range is very wide. So these bees, the capensis bees cannot move on their own. So this is a human facilitated spread of parasitic honeybee, and this has been shown by our colleague Vincent Deiterman, where he did the surveys between migratory and exposed apiaries and those beekeepers that are not sedentary. So if you look at the losses here, the losses are more in migratory beekeepers. So what happened with the capensis is, if you have infested colonies, you have to destroy them. Otherwise, they keep moving from one hive to the other in an apiary setup. So as at that time, it was recommended that a migratory beekeeping should be stopped for at least 1 or 2 seasons until the outbreak is over. But this was not the case, because beekeepers, you could understand, beekeepers have to move for pollination, and this is their livelihood. So as at that time, there were requests to look into other ways of solving this problem. As usual, scientists, you get requests from beekeepers. Despite that, it's a human thing. So what came to our mind is that to see, okay, since this is actually a chemical thing and it is based on pheromone, can we find a way whereby maybe increasing pheromones in the resident hive could help control or manage until the resident bees are able to raise another queen? So as we were discussing that, we know that in the literature, there is what is called the photoadjunctive response. In the queen honeybee, this is when the resident workers now raise a false queen. So a false queen is a normal worker bee that now takes charge of the hive before there is new queens. And then she has all the physiological presence of physiological characteristics of a queen, whereby she can bring retinues to her. And then, but what we don't know, what was known then, is if this is actually governed by pheromone. So what we did is with CNC, having now the advantage of the clone seated in between the queen and the worker pathway, to see if we can use exogenous pheromones to turn a normal worker bee into a false queen. So what we did was we took synthetic pheromones and we put it at the back of a bee, and then we took a dummy and then put a bee at the back of the bee without a pheromone, and we have a bee that is not that doesn't have pheromone. So we did this cage experiments, and we did both with the M. scutellata, which is a savanna bee, and also with the clone. So when I mentioned clone and when Fiona mentioned capensis, we should put at the back of our mind that we are referring to the clone here, not the endemic that is found in the Western Cape. So this was what we found. So the open bars are the false queens, and those that are carrying pheromones, and the closed bars are the workers. So what we found is that those that were carrying pheromones in both the scutellata, which are not known to produce a huge amount of the queen substance, they were able to produce a queen substance. And also in capensis, which are known to produce the queen substance, those that were carrying the synthetic pheromones produce more of the queen substance compared to those that are not carrying. This was quite intriguing. And then we look and see, okay, does that translate into the physiology as well? So we look at the ovarian activation. So this is a typical unactivated ovary. This is an activated ovary. So we look at so what we did then, we extracted the synthetic pheromone and also extracted pheromone from the capensis clone, because we saw here that they produce more. And then we did the same set of experiment, but in this case, we have the clone extract and we have the pseudoqueen extract. And these are, the red shows the fully developed ovaries and this underdeveloped. And then if we could look here, those that were carrying the pheromones, is now PCs, pheromone carriers, in both capensis clones and scutellata. You see they're the ones that show the development of the ovaries. So we were able to turn workers into false queens. But then we have to look into the application of that and that for. So now I hand over to Fiona to take to the second part.
Thank you, Dr. Yusuf. Thank you, Dr. Yusuf, for that good introduction to the topic. And also many thanks to the organizers for putting this meeting together and for the people who've made time to attend. So I'm going to present parts of the work that formed my thesis, my PhD thesis. And this will look at the genes involved in the biosynthesis of mandibular gland pheromones in capensis clones. And here I'll talk about the hydroxylation patterns in the mandibular glands, the transcripts involved in the biosynthesis of these mandibular glands in a larger transcriptomic study, and the effects of the host queen in the development of parasitism. And then Dr. Yusuf will come in and talk about Gemini in some work that he's done. So we've heard quite a lot about the differences that exist in the mandibular gland profiles of workers from various honeybee subspecies in Africa. And specifically here in South Africa, we've heard about the high 9-HDA production in capensis workers, predisposing them to parasitism. And this, for example, is part of a study that was done by Zeng et al. In 2010. You can see the capensis in the first 3 bars. These are capensis in the native region, and you can see they have a high proportion of 9-HDA and quite a bit of 9-ODA for a queen right worker. And the scutellata, for example, which is the open bar at the bottom, you can see it has produced quite a lot of 10-HDA, worker substance, and very little 9-HDA. So more of the worker compounds and very little of the queen compounds. So that difference is very interesting here in South Africa, South African honeybees. And even going deeper, just looking at clones specifically, the parasitic clones, we'll see further ahead that queen right clones are producing, clones collected from queen right colonies are producing high amounts of the precursor compound 9-HDA and very little of the 9-ODA, while clones collected from queenless colonies are producing high amounts of the queen substance, which is 9-ODA, and very little of the worker substance. So looking at this, our question naturally became, how are they able to do this? Because these are, morphologically, these are worker bees, but they're able to produce these queen typical acids. So we looked at the biosynthesis pathway, which Dr. Yusuf talked about a little earlier, starting with the entry-level biomolecule, which is stearic acid. It undergoes activation and acylation into stearic acyl CoA. And then this molecule, so this takes place in the cytosol, and it's transported into the endoplasmic reticulum where hydroxylation takes place. And hydroxylation takes place in a case-specific manner. So there's a bifurcation of the pathway here, as can be seen, and we see that omega -1 hydroxylation rather, in the penultimate position takes place in queens, leading to products that are going to be queen typical, such as 9-ODA and 9-HDA, while hydroxylation in the omega position takes place in workers, leading to formation of worker typical acids such as 10-HDA and the precursor molecule 10-HDA. So here we have various enzymes that participate in the formation of these molecules, and I'd like to highlight the cytochrome P450 enzymes that participate in hydroxylation. And after the hydroxylation products, when they get to the peroxisome here, where oxidation, incomplete beta oxidation takes place, we have oxidases, thiolases, and hydrolases. And the products of this, such as 9-HDA, are then oxidized to the queen substance, which is 9-ODA. And this part of the pathway here only takes place in queens. So when we looked at how these typically morphologically workers are able to produce queen typical acids, just looking at this pathway, the first place that we came to was the hydroxylation step. So we picked out 2 cytochrome P450s that have been shown in other studies to participate in omega hydroxylation, so that's in workers, and omega -1 hydroxylation in queens. And we'll see that right ahead. And our sampling looked something like this. We looked for colonies that were parasitized by the capensis clones, and these are scutellata colonies with capensis clone parasites. So where the host queen was present, we call that a queen-right colony, and that is basically sampling before the queen death here. And where the queen was absent, and absence of queen-laid brood, that was a queenless colony. So that was sampling after this part of the life cycle of an infested colony. So in some parts, we sampled adult capensis clones, and we also took the brood, took it to the lab, incubated using standard procedures, so that we could get bees that we could rear in cages and monitor that on an age-by-age basis. So we collected workers at day 0 for capensis clones, day 3 for scutellata, simply because of physiological developmental differences there. And then day 7 and day 10, these were called now the adults and the young ones. The young ones are day 3 and day 0, and then the adults are day 7 and 10. So we took them through the whole procedure that has been described before, where we extracted the mandibular gland pheromones. We extracted the mandibular gland pheromones for pheromone profiling. We checked on the presence of the spermatheca, very important as a diagnostic trait for the capensis clones, so the parasitic bees. And then we also looked at ovarian activation to see whether these bees were actually ready for parasitism, or actually taking the process of parasitism was already taking place or not. And we also had, of course, tissue that we used for the gene expression profiles. So what we found is that older capensis and scutellata workers were producing very different types of pheromone profiles. For example, the scutellata, if I start with the ovarian activation, for example, the older scutellata had majority stage 1 and 2 ovarian activation. That is inactivated ovaries. That is considered inactivated ovaries, and some bits of activated ovaries, that is stage 3. So they had just started activating the ovaries at day 7. The scutellata, the capensis clones, on the other hand, that's the parasitic bees, had about 30% that was stage 2 inactivated ovaries, but 80% in total stage 3, 4, and 5. So these are bees that were ready to lay eggs and continue the process of parasitism, bringing forth other capensis clones. And looking at the pheromone profiles, we saw that the scutellata were producing mostly worker type acids. You can see here 10-HDA, the proportion is about 75%. And the next compound in highest proportion was 10-HDA, the precursor molecule to 10-HDA. So basically they were continuing with the worker type profile and a bit of 9-HDA. 9-HDA also show of dominance, production of queen-associated pheromone components. The capensis were, it was the exact opposite, where a huge proportion was production of, that is about 75%, 9-ODA. That is dominance. And the next highest molecule was 9-HDA. So basically it was the queen substance and the precursor of the queen substance. Looking at the genes, the hydroxylation step catalytic enzymes, we find that the capensis clones were expressing a high amount of the queen-associated P450, that is CYP6BD1. In future, I can just call it 6BD1, a high proportion of that as compared to the older scutellata. And in contrast, the older scutellata were producing a relatively higher amount of the worker type P450, that is CYP6 is 8. So this might explain why the capensis clones are producing high amounts of queen-typical acids. They were simply upregulating the expression of molecules that are highly expressed in queens. Sorry, just 1 minute. Then looking at the younger bees. Looking at the younger bees, we see that there was no difference in ovarian activation. So both day 0 capensis clones and day 3 scutellata had stage 1 and 2 ovarian activation. That again considered not active. Those are ovaries that have not been activated yet. But when you look at the profiles, the pheromone profiles, you see that the younger capensis clones, that's the purple here, are producing 9-HDA, very high amounts of 9-HDA. That is at day 0. This for us was a very surprising finding, just showing that the capensis clones, even upon emergence, day 0 is less than 24 hours old or less than a day old. These clones are already producing queen-typical acids. At stage 3, of course, at day 3, the scutellata was still producing large amounts of worker-typical acids. And we looked at the gene expression profiles, and there was no significant difference in any of the genes that we looked at, in neither CYP6A8 or 6BD1 expression in either scutellata or capensis clones. And well, this is not surprising in itself because there are about 34 P450s that have been identified, and we only selected 2. These are the 2 that had been shown to be highly expressed in workers of other subspecies, but clearly here, maybe they're not what the capensis and scutellata start out with. So this took us now to the transcriptomics work, looking at the larger gene expression profiles. And first, I'd just like to mention that when we did a PCA analysis of all the transcripts with a non-zero read, what we found was that the larger portion of the variation in our data was due to subspecies. So the greater differences were between scutellata, generally scutellata and capensis clones, as you can see here, 52.8%, as compared to the age-related differences. So whether you're looking at younger versus older bees. So the subspecies held greater weight in the variation. And what we found from the transcriptomics data is that in all the stages of the biosynthesis pathway, from the beginning formation of stearic acid to the oxidation of 9-HDA to 9-ODA, these steps can be differentially expressed in the 2 subspecies and also in the different ages. So meaning these are steps that are technically, it's possible for them to be regulated by honeybee queens. So for example, we found 2 acetyl-CoA syntheses that were upregulated in the older capensis versus the younger ones. And this is key information of acetyl-CoA and the fatty acid syntheses and the stearic acid rather. We found 6 transcripts that are participating in the formation of stearic acid and also the acylation of stearic acid. Of these 6 transcripts, 5 were upregulated in capensis and 1 in scutellata, and that was in the older scutellata. And when we came to the P450s here, participating in the hydroxylation step, what we found was that I think we identified about 27 P450s that are participating in this step. And for the older capensis and even the younger clones, we found that more of the P450s that were upregulated were those that in other studies had been shown to be upregulated in queens that were either mated or virgin queens, or in workers that were from queenless colonies but with developed ovaries. And the reverse was true for the scutellata. Most of the genes that were upregulated in scutellata had been shown in other studies to be upregulated in queenless workers with undeveloped ovaries or in queen-right workers. So that's a big difference there in the hydroxylation pathway. It explains quite a lot. And another key thing for us was some of the rate-limiting steps. For example, transport of the hydroxylated molecules from the endoplasmic reticulum to the peroxisome where the incomplete oxidation takes place and all that. We find that the genes that were upregulated in the older capensis. So for example, here the acyl-CoA binding protein that's responsible for the transport is upregulated in the capensis. What this means is that we now have like a way of understanding why capensis clones are able to produce not only queen-typical acids, but also why they're able to produce them very fast, these queen-associated acids, and also why they produce such high amounts of these acids. So they are also upregulating these rate-limiting enzymes that are participating in very rate-limiting steps. And also I'd like to highlight here the alcohol dehydrogenases that are responsible for the conversion of 9-HDA to 9-ODA. We identified 4 that were differentially expressed and 3 were upregulated in capensis and only 1 in scutellata D10. So that's also something that gives us an understanding of why the D10 capensis, of course, are able to convert 9-HDA to 9-ODA. So the last thing that I'll talk about is the effect of the host queen in the development of capensis. So we've been talking about sampling from queen-right and queenless colonies, but the question then becomes, is the queen able to exert any effect on the development of parasitism in these clones, or does it mean that once a colony is infested, then it must go through this whole cycle of death of the queen and increase in the number of parasites and the decrease in the host number of workers and thus decline of the colony? So we sampled here adult workers only. So these were not caged experiments. These are clones that were collected from infested scutellata colonies. We got the adults and went through the whole process of assessing the pheromone profiles, looking at the presence of somatica and the ovarian activation stages. And what we found was that the clones that were collected from queen-right colonies, so where the queen was present, they were producing a high amount of 9-HDA, but very little 9-ODA. So they were producing the precursor to the queen substance, but were not able to convert that to 9-ODA, the queen substance. While the clones that were collected from queenless colonies in the absence of the queen, they're producing rather high numbers of 9-HDA, but even higher numbers of 9-ODA. And we used a ratio, this 9-ODA over 9-ODA plus 10-HDA. So looking at the proportion of 9-ODA in combination of 9-ODA and the worker substance 10-HDA, just to look at how queen-like a pheromone signal is, or how queen-like a pheromone profile is, rather. And what we came to see was that the queenless clones are producing majority, in fact, this is almost 1. It was, I think, 9.8 something, 0.98 something, this ratio. So they're producing majority queen-like pheromone signals. The pheromone signals could be described as queen-like, while the queen-right clones were producing majority worker-type acids. And you can see this is about 0.2, less than 0.2, in fact, for the majority of them. But there's a huge variation in this place here. There's a huge variation. And when it came to ovarian activation, what we realized was that the queen right clones had ovarian activation stage 1 and 2 majority, and about 30% was stage 3. So 30% had started activating their ovaries, while stage 1 and 2, that is all inactivated ovaries. And the queenless clones had a very small proportion that was stage 3, and the rest is stage 4 and 5, meaning that they were able to become parasites, fully become parasites, to the point some of them actually had already begun laying eggs. And this level of variation again goes with the variation that was seen in the classification of the pheromone profiles. So what we understood from that is that the queen-right and queenless clones are both using the queen's pathway. So if you look at this blue here and say that this is the worker pathway, you'd expect that workers in general would come and hydroxylate at the ultimate position and then come produce 10-HDAA and 10-HDA, the worker substance. But what we find in the clones is that most of them are, the queen is unable rather to control the hydroxylation of the capensis clones is unable to control parasitism at the hydroxylation step. So here we find that the clones are hydroxylating at the penultimate level and forming a lot of 9-HDA, as are doing also the queenless clones. But the queenless clones are able to activate this 9-HDA into 9-ODA. So ordinarily, we looked at what enzymes participate in the conversion of 9-HDA to 9-ODA. And from the work of Malka in 2014, we were able to identify the alcohol dehydrogenase that's responsible for this step. So we looked at the expression of this gene in the queen-right and queenless clones. And what we found was that the queen-right clones, as one would expect, had very little levels of the alcohol dehydrogenase, while the queenless clones had about 5 times the amount that could have been expected from, that were expressed in comparison to that of the queen-right workers. And of course, this now gave us a deep understanding as to why the queen-right clones were producing a lot of 9-HDA, but were unable to convert the 9-HDA to 9-ODA. Yeah, thank you.
Yeah, thanks, Fiona. Let me see how I can wrap this up. So Fiona had told us about all these enzymes that have been that are responsible for this for this conversion of the fatty acid. But one thing is that there's been a lot of work done on actually the genes that is responsible for this stereotypical pathogenesis. And one of them that I will highlight today, it's of the alternative splicing of a single transcription factor, which drives this teletoki. And this was work done by colleagues where they found they made this double exon model, which where you see the red one is showing the queen line, the solid dark is showing the arenatoki, which is now haplodiplody, and then teletoki, which is now the ability of workers to develop diploid individuals. So using this model, you could see that the presence or absence of the exon 5, it determines the sterility or the laying ability of the workers. So the good thing about this model is that you can make an isoform of this gene and using this alternative splicing and then see it effect. So what we did then was to have a look at the Gemini transcription factor, which is known as genitalia missing, and see if that has a role in the mandibular gland pheromone synthesis. So in the one, you have a control group, and then you just the group which you fed only with scrambled the siRNA, and then the other one with the Gemini. So what we found is that those that were fed with Gemini had more 9-ODA and then less 10-HDA, which shows that they are which shows the function of Gemini in the production and synthesis of mandibular gland pheromone. So now wrapping up the talk with what of other pheromone signals. So as I introduced earlier on, we have now heard of the mandibular glands and the synthesis, now the targal gland. So this was work done by Olabimpe Okosun, who was a PhD and postdoc in our lab some years ago. So what Ola did was to look at the targal gland secretion, especially in our bees, because the workers produce have a developed targal glands, and then look and see if there's a correlation between workers that are of the clones and if it has a function as that of the mandibular glands. So what Ola found was that looking at the capensis clones, most of the workers are queen-like, the 9-ODA, 9, and then most of us scutellata are worker-like. And then she looks at the targal gland pheromone bouquet, and voila, she found almost the same, where workers of capensis have more queen-like compared to worker-like. This was very interesting. When Ola was doing this work, she found out that there are some esters that were initially only reported as brood pheromones, but then she found them in this targal gland secretion. And then we were all surprised, and then Ola had to go and collect bees from the hive and look at the targal glands, and behold, she found them. Then we look and see if those also have both a primer or releaser effect, because now the question of redundancy arises. So what she did was she made blends. One of the blend is that of palmitic acid and the hydrocarbons, and the other only esters, and the blend 3, a mixture of those. And so if you look here, this is now the releaser effect. You see that blend 2 had more releaser effect. If you look at also the ovarian activation, you look at the blend 2 also had more bees that had ovaries developed. So Ola could show that the targal gland has the same function or act as a synergy with the mandibular glands. To sum up all the work, we recently had this review published where we put together the information we have available on the ovarian activation status, mandibular gland, targal gland, and Dufo glands. And with that, we could classify honeybee workers into different evolutionary stages, starting from a false queen, which is now a queen that has activated mandibular glands, activated ovaries, and activated targal glands, to a laying worker, which has a mandibular glands pheromone like a worker-like, but activated ovaries, and then also could lay eggs. From a normal worker without mandibular gland profiles and targal gland profiles of a worker and non-activated ovaries. And then there is this group that is known as the sapient queens. This has a mandibular gland like of queen-like of mandibular glands, queen-like targal glands, but then they have inactive ovaries. To sum up, we have shown that you could use exogenous pheromone sources to make false queens, and then that the capensis clones are primed from the beginning to produce queen-like workers, which is an advantage that they have when they use up colonies of other subspecies. And then we could show the different enzymes that are responsible for the omega-1 hydroxylation. And then we were able to show that under queen-right condition, the queens in a scutellata colony are able to try to fight the clone infestation. And then we could show that Gemini has a function in their role in mandibular gland production, and also that this control is not only from mandibular gland but multiglandular sources. And then we're looking at other glandular sources as well. So finally, I have to acknowledge colleagues and collaborators, Professor Christian Pegg, who is the director of our research group, Professor Robin Crew, who is the founder of a research group, Okosu, with colleagues from the lab that we've collaborated with from the Martin Luther University in Halle, and then colleagues here, and then funding from various organizations, starting from the Department of Science and Technology, South Africa, OWSD, African Union, the Humboldt Foundation, the University of Pretoria, and then the NRF. And then finally, thank you for listening.
Well, thank you very much both for a very nice and interesting presentation. While we wait for the questions of the audience, I have some personal questions to ask you based on the presentation you just showed us. The first one I have is Apis mellifera capensis considered a social species?
Yes, yeah, all these are social. So that's why we try to make the distinction between the parasite and the capensis bees. So the capensis is a bee, a normal bee. It acts as a normal bee when it is in its native range, which is in the Cape. It becomes a social parasite when it gets moved out of its native range. And the reason why it becomes a social parasite is this advantage that it has over other subspecies, because this is a bee where in its normal range, almost every worker is producing 9-HTA. And that is why it was very interesting from the findings of Fiona that actually, aside from the control at the hydroxylation part, which was what is known in the literature, there's actually another control down at the using alcohol dehydrogenase, which is stopping the conversion of 9-HTA into 9-ODA. Because the difference between our bees and European and other subspecies, that in other subspecies, the workers solely follow this omega hydroxylation. So you don't find the 9-HTA in other bees, only in our bees, because it's a puzzling and mind-boggling in that we're trying to see that waiting, there is another, is not in our bees, is not truly mainly a bifurcated pathway, because our bees can move between 9-HTA and 9-ODA, which is also an advantage to them when there is queen loss. So they can easily raise a false queen, which can take over before the queen is raised. I don't know if I've answered your question, Tasha.
Yeah, I didn't get this subtlety that it was when they were invading that they changed their behavior. So we have now some questions from the YouTube chat.
Okay.
I guess the first one is, the Cape honeybee seems geographically quite restricted despite its parasitism. Do you expect them to spread further north, or are there ecological constraints, for instance, climate?
No, due to the restriction, without human facilitation, the Cape honeybee would not shift its range. There have been studies looking at what is actually restricting them from the range. So if you, the geographic, there are two things that we think is restricting them. One is the geography. That introgression zone is usually in a place known as the Karoo. And the Karoo is such a vegetation that is not very friendly. It's a harsh environment. So it's one of the things that we think it's stopping them. Another thing is genetics. It is shown that the hybrid, because this teletoki gene is said to be lethal if it is expressed in a haploid condition. That's why you don't find them in drones. So that also explains why you don't find the facilitation of it moving beyond its geographical region. So the problem why it is a parasite is a human facilitated thing. It's not something that goes naturally. And it's highly unlikely that on its own it will move. So one thing about the clone is that this clone is, so when this outbreak was started in the 90s, it was reported that it was around 200 or 100 colonies that were moved, but among these, only one lineage established itself as parasite. So it's not the same lineage, it's not all the lineage lines that become parasitic. So up until now, we're trying to understand why is it that it's always this one lineage. That's why it's called a clone that is only able to establish itself when it's moved. So now we have a project with another colleague who are looking at sampling clones from different years to see if actually the clone remained the same or there is a change in its geographical, in its genetics. I hope I have answered that.
Yeah, that sounds very interesting. So there is a second question from the chat. What keeps rampant parasitism from causing colony collapse in the capensis colonies in their native range?
Good. That is a good question, and that goes back to what I had said earlier, and to this finding of Fiona that actually there is another regulation down in the synthetic pathway, which is that of alcohol dehydrogenase. So from the analysis we made, if you compare the pheromone production in terms of the queen pheromone between a normal capensis worker, that is laying, with the queen of scutellata, that capensis worker would have almost 10 times the production of a normal queen of Scutellata. If you compare the production of a capensis on its own with its queen, it is also a ratio of 10. Its queen produces more 9-HTA. So the theory we have is that the final step that is enabling the queen in its native range to control it. And then also we can look at the evolution of actually having this pheromone. So we have been postulating that why the Cape honeybee has this ability of producing queen pheromone. It's looking at the environment in the Cape. Is in terms of weather, it's not a very good environment, especially for bees, whereby it is prone to losing queens, especially in flight or something. And also there have been reported a colony resorption between the capensis itself. You can have an unrelated queen taking over one colony. So we think that the evolution of this pheromone is a trade-off or an adaptation to its native region. And then from a beekeeping experience, in terms of using stings and that, the sting of a capensis bee is much less painful than that of a Scutellata. So those are postulated theories that are not actually tested, but these I think are the mechanisms that the native capensis is using to keep the colony in check. Very interesting and lots of questions to answer.
Yeah, I think that relates to the question of one of our organizers, Dr. Abby George. He was asking whether there were strong behavioral differences between the workers from the different subspecies of Apis mellifera that correlated with the production of the 9-ODA or 10-ODA components, for instance, in terms of aggression.
Yeah, from personal experience working with the bees, yes, and from also other colleagues that have worked with the different subspecies. That is and they also up here, what we realize that in terms of usurpation is that since when clones gain access into the hive, they usually hide themselves. We often realize that those colonies that are large colonies, the capensis clones actually invade them most. In colonies that are not large, the workers are able to detect this foreign source of pheromone and kick them out. So there is that thing. So yeah.
Okay, and then he had a second question. Do we know what chemical signals the queen uses to control the conversion of the precursor to 10-HTA rather than 9-HTA in workers?
No, I don't know. Fiona, do you have what are your thoughts about that? We don't actually know, but Fiona, what are your thoughts?
No, we don't know.
Okay. A question for Yusuf, just a general question. Do worker honeybees produce sounds to communicate with each other and or with the queen?
Yes, they do usually produce sound as a form of communication within them and also with the queen. It's actually something that we actually use when we talk about communication in insect. Sound communication in workers, it's actually been shown to be possible. It's also something that has been used during the waggle dance as well. So the bees use they use sound to communicate.
Yeah, I guess in different other species of bees as well, the sounds, the buzzing is quite important in communication. Yeah, I had some question about the mechanism by which they parasite the other honeybee subspecies. Like, do they change their cuticular profiles to be accepted for the host colony, or?
Can you repeat that, Natasha? I lost you there.
Do you know by which mechanism they are accepted by the host colony? Because usually there is some recognition of like species or even different nests and hives.
Yeah. So now it's something that is it's quite interesting and it would tell you that this problem is actually facilitated by beekeeping practice. Many years ago, our colleague Vincent Detman did experiments where he set up cameras to look at how actually clones get into the hives. It is very, very difficult. The bees actually guard. So out of his observations, and it's tons and tons and hours of videos, it's actually a 1 in 1,000 times chances under apiary conditions. But then now the question is, how is it possible with beekeepers? So one of the things is that when our beekeepers move these bees from the cape up or up and down, they don't close the hives. And the hives have been put in close proximity with one another. So the theory is that the capensis now takes advantage of that and then move in between the boxes. So what happens is that you find this beekeeper with big beekeeping operation, 10,000, 6,000 hives, they move them, they go down for pollination and they bring them back. And because it's such a big operation, they just keep the hives in one place. So ideally, with the capensis problem, when you move your bees for pollination, you're supposed to keep them in sort of a quarantine, you see, and also inspect them to see, because you can visibly should see the symptoms of capensis clones. One of the systems is that you see a raised capping. If you see raised cappings, or if you see eggs laid haphazardly, you know that there's a capensis in there. So when you see that, ideally what you do is you keep that box in quarantine, far away from other hives. Otherwise, it now gives the capensis room to move from one, and it is highly facultative and highly contagious if you have it. It does not kill the colony instantly. It goes up until 6 weeks because this is a parasite that also needs resources. But once the hive are close together, then it's a recipe for disaster, because most of the beekeepers, when they find they have a capensis infestation, they're losing between 80 or 90% of their colonies.
I also think sometimes it could have something to do with stress, because these colonies have been moved, and the workers, normally these workers have been moved for monoculture, like for pollination of one single type of crop. Sometimes I also think the stress that the workers are normally undergoing at that period of time might possibly make acceptance of a clone a little easier than a typical colony. Yeah, just sheer stress. Yeah.
Okay, well, I have many more questions, but I think our time is up. Thank you very much for your presentation. I thought it was really interesting and really complete, like from behavior to genetics, it was, and the implications for evolution and so on, it was quite impressive. So thank you very much for your time.
And thank you very much also for organizing this.
Thank you very much. Yeah.